Hi, this is Dr. Claire, and today we're going to be talking about prokaryotes. Prokaryotes are a very diverse group of unicellular organisms. They're the most abundant organisms on the planet, and they're really important ecologically and also in human health and a number of other systems. So they're a pretty cool thing. They're also the oldest organisms on the planet. So life on this planet arose about 3.7 billion years ago. We have pretty good evidence from the fossil record of fossil prokaryotic organisms from that time. So these earliest organisms were these unicellular, very small prokaryotic organisms. And for a great chunk of the history of the planet, they were the only life form that was existing on the planet. So the first eukaryotic organisms, which is the organisms to which animals belong, didn't arise until about 2 billion years ago. So there was a big chunk of time when prokaryotes were the only ones that were here. And they have been very important to the evolution of the planet, particularly because the first organisms that produced oxygen in the atmosphere were actually these prokaryotic organisms. So an organism like cyanobacteria that was able to photosynthesize and take in carbon dioxide, turn that into sugars, and excrete oxygen, was the first organism that was adding oxygen to the atmosphere. So the early atmosphere on the planet was pretty much devoid of free oxygen, and it took until we had these photosynthetic organisms that started to produce oxygen that we had a lot of oxygen in the atmosphere. And until we had a significant amount of oxygen in the atmosphere, the rest of life, the multicellular life, organisms such as ourselves, couldn't really survive. So it was really important for the building up of that oxygen that we had these cyanobacteria that were starting to photosynthesize. So the prokaryotes are a very, very diverse group. Many of them are probably unknown or undescribed. Since they're very small, they're microscopic, they're very hard to see and figure out what they are. They are hard to identify as well. And in addition, some of them have very specific growing requirements. So if you wanted to try to identify these organisms, it would help to be able to grow them in a lab. And many of them are very, very difficult to grow in the lab. They just won't grow under laboratory conditions. They only grow in the environments in which they normally live. There's also 2 domains. So there are 3 domains of life. The eukaryota, which is the multicellular organisms, and some unicellular ones as well. And then there's 2 prokaryotic domains. So the eubacteria, sometimes they are known as just the plain bacteria. Eu simply means new, so when they regrouped them, they called them the eubacteria. And then also the archaea. They used to be considered to be part of the same group, but they've discovered that there are some pretty significant differences between the eubacteria and the archaea, so they're now considered in different domains. So that's our largest taxa that we look at. So there are only 3 domains on the planet, and 2 of them are prokaryotes. Okay? The archaea are actually kind of cool because many of them are extremophiles, meaning that they're able to live in very harsh conditions where you wouldn't necessarily expect life to exist. So there's some pretty interesting organisms in that group as well. They are everywhere. They are in the air. They're on every surface. There are prokaryotes everywhere. In fact, in the human body, our human cells are outnumbered 9-to-1. So for every 1 human cell, you have 9 bacterial cells somewhere in your body, on your skin, in your intestine, in your lungs. There are bacterial cells living all over you all the time, and many of them are actually beneficial and not harmful in any way. So that's our kind of what we would call our microbiome. It's the ecology that lives with us all the time. All right, let's talk a little bit about what these prokaryotes look like. They are unicellular, so they have, just 1 cell. They do not have a membrane-bound nucleus or any other membrane-bound structures within the cell. So the DNA, which we would call the nucleoid, is found in a circular structure, and it's just kind of clumped together in a cluster in the middle of the cell. It's not segregated off from the rest of the cell. They do have a plasma membrane, like all cells on this planet, and then often they have a cell wall and then a sticky outer section called the capsule. They can move, many of them. They have flagella. Flagella are like little propellers that let them move through liquids. And some of them have what are called pili, which are these little extensions of the cell membrane that can allow them to adhere to structures and also sometimes to communicate with each other. Within the cell, aside from that nucleoid and some ribosomes that are just kind of floating around in there, there's not a lot of other cellular structures. So the eukaryotic cell is much more complex, and we'll talk about that in the next lecture. So as I said, these organisms are unicellular. They can, however, form what are called biofilms, where many individuals are associated together and kind of cling together. One great example of a biofilm is plaque on your teeth. So you might notice after you haven't brushed your teeth all day that you get this kind of white film on the surface of your teeth. That white film is a biofilm of bacteria that are living in your mouth and feeding on the remnants of the food that you have eaten. So they're getting all the food that they need from the stuff that gets stuck to your teeth, which is why you have to brush your teeth fairly frequently, because those bacteria will eventually produce acids, which then can corrode your teeth and cause tooth problems. They are very, very small. Prokaryotic cells are much smaller than eukaryotic cells. So this illustration right here, this is a white blood cell. White blood cells are part of our immune system that are largely responsible for dealing with prokaryotic cells. And it is engulfing and consuming some prokaryotic cells, which are there in green. So you can see they are much smaller than that white blood cell. Again, there's no membrane-bound nucleus. They have that circular strand of DNA that's just kind of clumped up into what we call the nucleoid, and they can only reproduce by cell division. So they can't have sex, and we'll talk about that a little bit more in a minute. All right, so let's talk about the difference between the bacteria and the archaea, those two different domains. A lot of the differences are very, very technical, and because we don't expect you to have had a strong background in chemistry yet, we're not going to go into it in too much detail, but some things to know that the lipid structure in the membrane, the cell membrane, is different. You can see in this little diagram here, there's this ester group here that's not there. So that's a main difference. You don't have to know what that means. Just know that the structure of the lipids that make up the cell membrane is different between the bacteria and the archaea. The archaea do not have a peptidoglycan wall. Peptidoglycan is a compound that many in the bacteria group use to make their cell walls, and the archaea don't have that particular compound. The DNA replication process is also slightly different between the bacteria and the archaea. And the archaea's DNA replication process is actually much more similar to the eukaryotic replication process. And also the gene expression, the way the nucleotides within the DNA are expressed into proteins, is slightly different between the bacteria and the archaea. And again, the gene expression is more similar in the archaea and the the archaea and the eukaryota than it is in the bacteria. Okay? So what that tells us, those similarities between the archaea and the eukaryota tells us that those two groups are more closely related. So if you look at a tree of the three domains that we have on the planet, you'll see that archaea shares a more recent common ancestor with eukaryota or eukarya than they share with the domain bacteria. So the archaea and the eukarya are actually sister taxa, whereas the bacteria are more distantly related. Okay? Okay. Because the prokaryotes are very difficult to identify, one feature that has been used historically is the shape of the cells. And this often comes into account when we look at the names of some of these organisms. So there's three basic shapes. There's bacillus, which are rod-shaped or kind of pill-shaped cells. There's coccus, which is round spherical cells, and there's spirillum, which is a spiral shape. Okay, so if you have ever heard of, say, lactobacillus. Lactobacillus is the bacteria that you use to make yogurt. Lacto means milk. Bacillus means pill-shaped. It's a pill-shaped bacteria that likes milk. So lactobacillus. Or streptococcus. Streptococcus is it's a bacteria that causes strep throat. and it's a coccus-shaped bacteria. So a lot of times those shape terminologies show up in the names of the bacteria. Many bacteria or prokaryotes can form what are called endospores. And the endospore is a structure that allows the prokaryote to survive very harsh environmental conditions. So it's a very thick wall that protects the genome and allows that organism to survive desiccation, dehydration, really hot temperatures, poor growing conditions, whatever environmental stressors it might encounter. It allows them to survive that, and then when the conditions improve, then it can come out of the endospore and grow and reproduce again. So it increases the survival in harsh environmental conditions. Now, this is important for some groups that you might have heard of, say tetanus. When you step on a rusty nail, you are in danger of getting tetanus, so you have to go get a tetanus shot. Well, the tetanus bacteria can actually survive on the surface of that nail for long periods of time with no moisture and not much to eat. And then when you step on that nail, that bacterium gets into your tissue, and that's a great place for it to grow. There's lots for it to eat there, and it's very happy, and then you get sick. Another really great example is botulism. So botulism comes from a bacterium that eats food. So when you can food, you have to heat that food to a certain temperature, and the botulism can go into an endospore and actually survive that heating in some cases. If you don't heat it enough, some of those endospores will survive, and then when you can the food, once it cools back down, they can hatch out and start to consume that food that's in the can. When they consume the food that's in the can, it produces gas, and the can gets bulgy. This is why you should never eat canned food where it's bulging out. If it's dented in, it's probably okay. If it's bulging out, don't eat it. It's very dangerous. And then a final one, anthrax. You may have heard of these anthrax scares where someone gets an envelope that's full of white powder. That white powder is the endospores of the anthrax that are dormant and just waiting. And then when you breathe in that white powder into your lungs, the anthrax spores are then in an environment they really like. They can grow and reproduce and make you sick. Okay? All right, so I mentioned earlier that prokaryotes cannot reproduce sexually. They only reproduce through cell division. So when you go through cell division, you make clones of yourself. You make individuals that are exactly identical to you. So one problem then would be, how do you get new genetic information into a cell? So in animals and plants, we have sexual reproduction where you combine genetic information from two individuals, and that increases genetic diversity. So how can a prokaryote increase its genetic diversity? Well, they can have what's called horizontal gene transfer, and that's when you get a genetic material from another individual, not through reproduction, but just through picking it up in some other way. So it's basically like if I walked up to someone and I went, pook, and I poked him, and I transferred some genetic material to that other person. It's another individual. We didn't make babies. I just gave them a gene. Okay? So that's what we're talking about with horizontal gene transfer. Vertical gene transfer is transfer from a parent to an offspring, and so that's a different type of gene transfer. We're talking about a gene transfer from one individual to another, which is a little bit weird, and animals don't do that. So it's something that the prokaryotes do that's a little bit unique. And there's actually 3 different ways that prokaryotes can have horizontal gene transfer. There's conjugation, transduction, and transformation. We'll talk about each of these in a little bit here. All right, so conjugation is, this is the closest thing that prokaryotes have to sex. So in conjugation, one prokaryote individual will have a particular plasmid. A plasmid is a little bit of DNA that's not in the main genome. It's a little separate little circle. And that plasmid will allow that individual to grow these pili that come out that they can contact another individual. Okay, it's usually called the F+ plasmid because that stands for fertility plasmid. It allows them to grow these pili. So that plasmid will allow them to grow the pili, touch another individual, and then they will transfer a copy of that plasmid through the pilus to the other individual, and then that other individual also has the F+ plasmid, and it can make its own pili and continue to transfer that thing. So it's a way of transferring these little plasmids back and forth between individuals. Okay, so that's conjugation. Transduction is actually kind of an accidental transmission of genetic material through viruses. So there's a lot of viruses that prey upon prokaryotic cells. And so when a virus attacks a prokaryotic cell, it attaches to the cell and it injects its DNA into that cell. That DNA then hijacks the cellular mechanisms of the cell and uses that cell to build more viruses. And it chops up the host DNA in the process. But then when the viruses get built, some of those viruses, as are indicated here by these little capsules with blue in them, some of those viruses are going to pick up the host DNA, not the viral DNA, just by accident. And then when those viruses go to a new cell and they inject that DNA, they're injecting prokaryotic DNA from the previous host into the new host. But that's not viral DNA, so it's not going to harm the host. And sometimes that prokaryotic DNA gets incorporated into the new host's genome and remains there. So it's one way that this DNA can get back and forth between these cells. Okay? All right. And then the last one is transformation. And transformation is kind of cool because it's a response that prokaryotes have to being under stress. So if they're in a slightly stressful environment, their cell membranes actually become somewhat permeable to chunks of DNA. So if another cell were to die, another prokaryotic cell dies, what happens is that cell will rupture and there'll be little bits of DNA kind of floating around in the media, so in the water or whatever. And then the other cells, if they're under a little bit of stress, they may make their membranes more permeable and they may pick up a piece of that DNA. Now, why would you make your membranes more permeable if you're under stress? Well, if you're under stress, you're having a hard time coping with your environment. It might actually be beneficial to pick up a random piece of DNA because that random piece of DNA might have a gene on it that allow you to survive in that somewhat harsh environment. So they pick up these chunks of DNA, they get incorporated into the genome, and then they're in there from then on. We actually make use of this as humans to, it's called molecular cloning, to get genes that we're interested in into a bacterium, and then we can grow a bunch of that bacteria, particularly usually we use E. coli. We grow a bunch of that bacteria, and then we have a bunch of copies of that gene, and we can get a bunch of the gene product as well. So it's something that is used a lot in lab. All right. The next thing that's really important is to talk about cellular metabolism. So in particular, where do these prokaryotic cells get their energy, and where do they get their carbon? And when we're talking about carbon, most organic molecules are carbon-based. So what that means is that the carbohydrates, the fats, the proteins that make up your body, they all have a carbon backbone. Usually the base carbon-based molecule is going to be these carbohydrates. So where does the organism get its organic molecules from, and where does the organism get its energy from? So let's start with the energy. Generally, there's two ways that organisms can get energy. They can get energy from the sun, or they can get energy from energy that's stored in other chemical compounds. Okay, so if you get energy from the sun, you're called a phototroph. You're getting your energy from the sun. Photo is light, so light energy. If you're getting it from other chemical compounds, you're called a chemotroph. Okay, so that's the first division. Now within each of those, you need to know where you're getting your carbon from. So either you can make your own carbon-based molecules, or you have to get them from somewhere else. You have to eat something else to get those carbon-based molecules. So if you can make your own, you're an autotroph. You make your own carbon-based molecules. If you can't make your own, you're a heterotroph. That means you have to eat something else to get those organic molecules, okay, to get those carbon-based molecules. And so each organism that you look at, you can combine these terms. So you have photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs, okay? So those are the four different categories, all right? So your photoautotrophs, those are your photosynthetic critters. So things like plants, but there's also cyanobacteria, algae, things like that, things that are green generally, and they're trapping light and they're making organic compounds, okay? You can also have photoheterotrophs. A photoheterotroph can trap light energy, but it can't make organic compounds. So it can still use that light energy for other things, but it still needs to make its own organic compounds. Or not make, it needs to eat something to get its own organic compounds, okay? And there are some prokaryotes that are photoheterotrophs. Prokaryotes are the only things that are photoheterotrophs, okay? Now on the chemotroph side, if you are a chemoautotroph, that means that you get your energy from breaking down some sort of chemical compound or putting together chemical compounds, and you can take carbon dioxide and make these organic molecules yourself. So these are things that can make organic molecules. They can fix carbon, but they don't use sunlight energy to do that. So there are sulfur oxidizing bacteria that use sulfur compounds to get their energy. Those are the ones that tend to live by deep sea vents. I'll show you a picture of that in a minute. There's some hydrogen bacteria that can do this as well. And only bacteria are chemoautotrophs, okay? And then you have your chemoheterotrophs. Chemoheterotrophs have to get their organic compounds from somewhere else. They have to eat something. That's why they're a heterotroph. And a chemo means that they get their energy from breaking down chemical bonds. Most chemoheterotrophs get their energy from breaking down chemical bonds in the food that they eat. So for example, animals are all chemoheterotrophs, so are fungus, okay? And there are also some bacteria that are chemoheterotrophs as well. Okay, so let's take a look at one of those chemoautotrophs. There are these chemoautotrophic prokaryotes that live near these hydrothermal vents deep in the ocean, and these hydrothermal vents are pumping out a lot of sulfur compounds and a lot of heat, and the chemoautotrophic bacteria can take that energy from those compounds that are coming out of these vents and use it to make organic compounds, to fix carbon into things like sugars. And then once they are there doing that, then there are lots of other organisms within those deep sea vent communities that are going to be getting their energy from those chemoautotrophic bacteria. So, for example, there are these really cool tube worms, and there's some weird clam things, and all of it is based on a ecosystem where the base is these chemoautotrophic bacteria. On most other ecosystems on the planet, the base is something that's photosynthetic, something that's trapping sunlight energy and using it to fix carbon. But in the deep sea vent ecosystem, it's not sunlight that's the source of the energy. It's these hydrothermal vents, which is kind of cool. All right. Okay, let's talk a little bit about prokaryotic ecology. Prokaryotes are actually extremely, extremely important to the functioning of the ecological systems on the planet. And the big thing that they do is that they fix nitrogen. All right, so a lot of the nitrogen that's available on this planet is found in the atmosphere. The atmosphere is about 80% nitrogen gas. But nitrogen gas is two nitrogen atoms with a triple covalent bond between them. So nitrogen gas is extremely difficult to break apart into individual nitrogen atoms. And so it's not really accessible or usable for most organisms. They can't use nitrogen gas to make nitrogen-based compounds like proteins. So there are these prokaryotes that can fix nitrogen. They can actually break that triple bond and then turn that nitrogen gas into things like nitrates or nitrites, or ammonium. And those are things that are then biologically available for other organisms. So it's a really, really important feature of these prokaryotes. And a lot of times they'll live in very close association with things like plants. So if you've ever heard about crop rotation, where you rotate between a legume like a soybean and the corn, the soybean has a symbiotic association with a nitrogen-fixing bacteria that is taking nitrogen from the air and turning it into this useful nitrogen. So it actually increases the nitrogen content in the soil, and then you can grow something like corn that doesn't have an association with nitrogen-fixing bacteria, and you don't need to fertilize the corn as much because the bacteria that was associated with that bean has already added nitrogen to the soil for you. Another really important thing that that prokaryotes do in the ecosystem is that they're decomposers. As you probably know, bacteria like to grow on things that are dead, and they will break down dead plant material, dead animal material, poop into structures, more simple structures that other organisms can then uptake and use use again. So they're very useful in terms of decomposition as well. We have our own prokaryotic ecosystem that live in our body. Some of these things are very beneficial. They can aid in digestion of our food. They can break down compounds that we can't break down ourselves. Vitamin K is actually an essential human vitamin that we can't produce ourselves, but it is produced by prokaryotes that live in our gut. And they can help us to fight off infections by harmful prokaryotes by outcompeting them. So I had an appendicitis and my appendix ruptured. And so it spilled the contents of my gut basically all over the inside of my body. That's very bad. Bacteria growing in places where it shouldn't be. And so I had to be on IV antibiotics for like 3 days. It killed all of my whole ecosystem. And the only thing that was left was this antibiotic resistant Proteus, which is a flagellated bacteria that can swim, and it just kept on showing up in all these different places, and I couldn't get rid of it because I didn't have any of my beneficial ecosystem to help fight it off. So I ended up taking a bunch of probiotic pills and stuff like that to repopulate my ecosystem, and I was finally able to get rid of this nasty Proteus that was kind of just taking over things. All right? Okay, so bacteria are also good in the human ecosystem. Of course, there are harmful bacteria as well. Bacterial diseases used to kill 20% of children before the age of 5. And with the advent of modern antibiotics, we have a much better control over a lot of these bacterial diseases, and also modern sanitation like proper sewage that prevents the spread of some of these diseases has also really helped reduce that. But there are still bacterial diseases that come and go, but there's many of them that you may know, like cholera and strep throat, and there are a number of different bacterial infections that are pretty serious as well. Okay? So sometimes prokaryotes are good, sometimes prokaryotes are bad. All right? That's the upshot. One thing that I think is particularly interesting is our use of antibiotics has actually caused evolution within prokaryotic populations. And if you think about it, it's not that surprising, because if you have a population of prokaryotes in your intestine or in your body and you take antibiotics, if you have one individual who has a mutation that allows it to resist that antibiotic and it kills off everything else, then you stop taking the antibiotic, that one individual who survived is going to reproduce, and all of a sudden you have a huge population of antibiotic-resistant bacteria. And so this is why it's really important to use antibiotics responsibly, not take them when you don't need them, because it's probably just going to cause more problems than it solves. And be aware, like we use a lot of antibiotics in agriculture as well, and so that can be really problematic if those antibiotics are causing a lot of antibiotic resistance at like say these commercial pig and cow farms and things like that. All right, so that's my lecture on prokaryotes, and I'll catch you next time.